Chapter 9, Section 2 talks about genetic crosses. And before we get to the crosses, we need to know the difference between a few terms. The first terms are genotype and phenotype. A genotype is the genetic makeup that an organism has, so the actual genes that it has. So for a particular gene, you remember you have two alleles for every gene. So you could have two dominants, you could have a dominant and a recessive, or you could have two recessives for a particular trait. The phenotype is going to be the physical appearance. So you don't always see every single allele that an organism has. For instance, in mouse fur, let's say black is dominant over white. Well, you could have a black fur and you could be dominant, or you could be dominant recessive. So you could have two different genotypes and still have the same phenotype. The only way you would get that recessive phenotype is if you had both recessive alleles. A couple other terms you'll need to know the difference between are homozygous and heterozygous. Homozygous means that you are a purebred. You have the same alleles for a particular characteristic, and you can be either pure for the dominant characteristic, in which case we call you homozygous dominant, or you could be homozygous recessive, where you have both of the recessive alleles. Heterozygous is also called a hybrid, and that just means that you have different alleles for a characteristic, so you have a dominant allele and a recessive allele. Now, probability is just the likelihood that a specific event is going to occur, and we use these a lot in genetics. Mainly, we can calculate these with Punnett squares. This example here is a Punnett square. On the top, you put one parent's genes, and on the side, you put the other parent's genes. Doesn't matter which one goes where, just as long as you have both parents on there somewhere. Then, the offspring are going to be filled in the four boxes. So the combination of those parents' alleles get filled in, and that can give you the possible ratios for what their offspring would have, both in terms of genotypes and phenotypes. Now, Punnett squares can be either in the form of one characteristic being crossed, which is called a monohybrid cross, and that would be one like down here where you've just got your normal four boxes and your parents on the top and the side predicting your offspring. You can also have a dihybrid cross where you cross two characteristics at once, or a trihybrid cross where you cross three characteristics at once. This is a sample of a dihybrid cross, and as you can see, it's a larger diagram than just your typical monohybrid cross. For this one, they happen to be crossing the seed shape and seed color for peas at the same time. So instead of a 2x2 box, they're doing a 4x4, and you can see that the parents have four alleles here instead of just two. They have two for one trait and two for another trait. In this situation, the round shape is dominant to recessive, is dominant to the wrinkled, which is the recessive. So here we have a heterozygous person for our P for this round shape. It has a dominant and a recessive. And then we're also heterozygous for the second trait, which is dealing with color. So yellow is actually dominant over green. And both of these parents happen to have the same genotypes here. Now, what you need to do with dihybrid crosses is then figure out what's going to be going in your top here for your female and your side for your male. Now, for doing two traits at once, you need to make sure that you have two letters being put above each one of these boxes instead of just the one, like we did with monohybrid. So, for instance, here we could get passed down a big R and a big Y. You could get passed down a big R and a little y. You could get a little r and a big Y, or you could get a little r and a little y. Those are all the possible gametes that this particular P could contribute to its offspring. You would do the same thing for the dad here, and then filling in the boxes is the same as your monohybrid crosses. You're just combining these two parents' alleles. So we would have big R, big Y, and so on all the way until you have your entire Punnett square filled out. A test cross can be used when you know the phenotype is dominant, but you don't necessarily know what its genotype is. Could it be two dominants, or could it be dominant and recessive? So what you would do is cross it with a homozygous recessive organism. So one that is pure for the recessive trait. Now, if you look here, you've got, we've tried out both of these possibilities, and with the pure dominant trait combined with the pure recessive trait, all the offspring would still have that dominant trait. There would be no possibility of getting the recessive trait with that particular combination. But if you had a heterozygous purple plant with the homozygous recessive plant that we would do for our test cross, then you would have a 50-50 chance of getting either purple or white. So if we got white flowers with this cross, we would know that the purple plant had to have been this heterozygous version, not the homozygous version. So that's called a test cross. Now, not all crosses are as straightforward as these ones. There are some different complications. A few of them are incomplete dominance, codominance, multiple alleles, pleiotropy, polygenic inheritance, and epistasis. With incomplete dominance, that means that both of the alleles are going to blend to make the phenotype when you have a heterozygous organism. So, for instance, in this particular type of plant, we've got red flowers combining with white flowers. Red is big R, white is little r, little r. Now, when you combine those two plants, you get an F1 generation that is all big R, little r. And those plants all come out pink. That means that we've blended the red and the white. So that is a form of incomplete dominance where you get that blending of alleles. If this was just our normal dominant recessive cross, then this F1 generation would all be red because it has at least one of these big R alleles. But in incomplete dominance, you're going to get that blending to show through. Codominance is similar. You'll need to know the difference between incomplete dominance and codominance. In codominance, both alleles are actually going to be expressed in a heterozygous offspring. So it's not blending, but you do see both of the traits. So, for instance, in this one, we're dealing with, horses or cows. And when you see those, the kind of reddish colored ones with also some white spots on them, they're a combination of the two. So when you mix one that's pure for that red color and pure for the white color, you're going to get the heterozygous version, the dominant and the recessive. Now in this situation, it's called roan when we see both the red and the white. So we don't see pink cows and horses floating around out there. There's no incomplete dominance, it's codominance where you see both of those characteristics from each of the alleles. Multiple alleles are traits that are controlled by 3 or more alleles for the same gene. So far all the ones we've talked about have been controlled by 2 alleles, a dominant and a recessive. Our blood groups, our ABO blood groups, are formed by multiple alleles. You could have the dominant A, which is represented by a capital I and a superscript A here. You could have a B allele, which is also dominant. It's a capital I with a capital B superscript. And you could also have the recessive allele, which is a lowercase I that they use to represent that. So there's 3 different alleles that can be combined, still in pairs, combined in pairs, but to give us all of our different blood types. So those are multiple allele traits. Pleiotropy is when one gene can affect many characteristics. So, for instance, the example here is the hemoglobin sickling allele. These are what normal red blood cells look like, kind of roundish, have this little, almost like a thumb imprint in them. And these are ones that have sickle cell. So they have these sickle shapes, they have these pointy ends. And what happens is that when a gene causes this sickle cell trait, the cells can then end up breaking down other red blood cells. They can end up clogging blood vessels, and all of these different things can occur from that: anemia, heart failure, brain damage, spleen damage, kidney failure. All these different characteristics could happen based on this one gene that causes these sickle cells. So that's pleiotropy, when one gene affects many characteristics. Polygenic traits are those that are determined by multiple genes, and many of our human traits are actually polygenic, which means that just not one gene controls it, but there's multiple genes that control this. For instance, skin colors. Okay, that's the diagram that I have up here. Each gene actually will contribute a certain amount of melanin. Melanin is what allows your skin to darken. It's a pigment. So each gene contributes a certain amount of melanin. So the more melanin you get, the darker your skin tone can be. So that's why we can have such a wide variety. Same thing with anything that we have a variety for. Our heights, our weights, our hair color. You know, we have these varieties. We're not one option or the other. With our heights, we're not all 5 foot tall or 6 foot tall. There's a variety. So there's multiple genes that are controlling and contributing to that particular characteristic, and that is a polygenic trait. Finally, there's something called epistasis, which means that the expression of one gene actually depends on another. So, for instance, with this one, I've given the example of hair color and albinism. You might have particular characteristics for maybe brown hair or black hair, whatever shade hair. But if you also have the gene for albinism, then that albinism gene is basically going to take over and overpower the other ones. So you're actually going to end up with that white color, like these mice here. If they have the albinism gene, it overpowers what genes it currently has and is not going to be able to display those ones. They're going to instead have that white color and those red eyes that are typical of an albino.